Starting up here in Biology of Fishes, today is October 30th, a Tuesday, and got a sunny day. We're all high and dry in Texas, and feeling for folks that are not so fortunate up in the Northeast today with that storm system up there. Being a Tuesday, today is lab day in College Station. And today, this afternoon, after this session, we're going to address Lab 5. And I've been working on that. It's a good lab. They're all good labs, Allison. That was my favorite, but somebody said one time, Dr. Neal, you say that every time, it's your favorite. And they all are, but they sort of build, and this last one is really my favorite. It deals with behavior. But before we deal with that, I want to finish a consideration of adaptation on the part of the individual organism to its time-varying environment. And I generally class those kind of responses as what I call acclimation or acclimatization. I'm going to just pace through here till we get to where we stopped last time. And I think it was at the point of dealing with the transient state changes. I sort of dealt with steady state physiological status upon compensatory changes in the organism. And now I want to consider how animals get from one state to another, which is, as usual, a lot harder to understand, and there's been a lot less attempt, I think, at that understanding, but it really is relevant to the real world because the real world is a changing place. So the first thing I'll deal with is resistance acclimation here. And the image that you see on the screen is a rather classic graph produced, I think, by Raleigh Brett, showing the acclimation state achieved as measured by heat resistance as a function of time subsequent to a step from one state to which the animal was fully adjusted to a different level of the same factor, temperature in this case. So he did transfer experiments from T1 to T2 and T2 to T3 and T3 to T4, and then also the reverse transfers. And what he measured was pretty clever, really. He said, well, I don't know how to go in and do enzyme assays and do whatever it is that I would need to do to actually record the changing physiological components of the system. Not even sure which ones are important. But what I do know how to do is measure emergent properties. And so what Brett did was make the transfers and then at intervals through time pull samples of animals out of the system and measure their ability to resist upper lethal temperature death, and compare that with the times that animals of known adaptation could resist the same temperature change. And by doing that, he was able to actually back-calculate the acclimation state that the animals had achieved. Does that make sense? So, if you got a whole bunch of animals that are steady state, and you know they're steady state, and you test them at a given upper lethal temperature and record their resistance times, and now you take your unknown group from your step change experiment after time t, here on the x-axis, and you expose them to that same level of the lethal factor, and you look to see how long they resist death, and then you say, well, whichever steady state group is the same as my sample defines the acclimation state of my sample. So you're kind of using an emergent property to measure the status of the system. And when you did that kind of experiment, this is the kind of result he got. So animals that were moved from T1 to T2 initially, of course, had acclimation states that corresponded to steady state T1. And then after a long enough time, their acclimation states shifted to those that would be steady state for T2, as they'd have to, right? And how long did it take? Well, it took a long time. It took, about 3 weeks for this lowest step. And I think the low step, by the way, was 4 degrees to 8 degrees. And I think these were goldfish. So I think T1 was 4, T2 was 8, T3 was 12, and T4 was 16. I think. But don't hold me to that. That's not the important thing. The important thing is for the lower interval, it took a long time. And initially there seemed to be some lag or latency in the response, where you can sort of see the physiological system kind of getting itself organized, and then it takes off, and then goes up in a sort of an exponential way to finally become steady state at the new value, T2. When you do the step from T2 to T3, you get the same kind of result, except it happens quicker, maybe only a week or so. And then from T3 to T4, it's really quick. And the initial latency there is hard to see if it even exists. It's almost like a pure exponential decay for that highest value. It only takes 2 or 3 days for the final transfer to reach steady state or near steady state. When you do the reverse changes, come on in. When you do the reverse change, it looks like the same kind of pattern, except the time it takes is longer for each of the intervals. And I've only drawn the one up here for T4. Notice something. Notice that, even though the T4 to T3 step doesn't seem like it's got the latency, the lag, the initial lag, the time it takes to reach steady state going down from T4 to T3 looks to be about the same as going up from T3 to T4. From T2 to T3. Going down from T4 to T3 is about the same as T2 up to T3. Why do you suppose that might be? Well, might it be the fact that in both cases, the animals are having to do their physiological business at a common temperature, T3. In other words, it may be that the temperature where you are living as you acclimate is the temperature that's driving the rate of the process. So animals going from T4 to T3 take about as long as animals going up from T2 to T3. And if you'd have looked at the others here, you'd get similar results. For example, T3 to T2, you can sort of anticipate, complete the set. And if you had to draw another line here, you'd probably draw it like this, where it comes down and comes to equilibrium at about the same time as going up from T1 to T2. So Brett, I think, showed us 3 things. And the first of these is that acclimation is faster over the higher of 2 spans of temperature that are of equal length in terms of measure degrees centigrade. For example, 4 degrees. So you acclimate faster from 4 to 8 than you do from 8 to 12, or 4 to 8 versus 8 to 12, for example. The other thing he showed us is that acclimation over the same span is faster than going up than it is going down over that span. And the final thing was that the general shape of the relationship or the pattern is sort of a quasi-exponential decay, but it's got this, little bit of a problem here at the beginning, like it's not starting out exponential. It's starting out kind of sigmoid. But in general, it's sort of an exponential decay process, it looks like. So I did some experiments like that. Yeah, Chris.
I was just going to say, you could have that sort of shape going in both directions, right? Like,
let's back up.
Would it be the same, as you were getting cooler in temperature? Would the rates be quicker?
Yeah, well, I was saying there, and I'm gonna go out of this, and since Chris asked the question, that you'd have the same kind of thing going down as going up, and I'm gonna do Alt Z, and I'm gonna try to draw on my graph here. I'm gonna draw what happens if you move a fish that's fully adapted at T3 down to T2. And what I predict would happen, consistent with the pattern that is shown here, is something like this. Whoops, where it would, well, I guess I got the wrong. Let me see if I can undo that and go back and change this to a pin. I think I had an arrow selected. Okay, what I want to do here is draw a smooth exponential decay with a lot lower rate that comes to equilibrium out here. And you can see my intent there. Those rough places are just shaky hands. But anyhow, the idea here is to draw a slower exponential decay where it comes to steady state at about the same time as T1 going up to T2. About here. So these two curves both reach steady state out here somewhere around 3 weeks. These two curves both reach steady state back here somewhere close to maybe 10 days. And if you imagine a T2 to T1, you couldn't even get that on your graph. It would be way out here someplace. I don't know how long it takes because I don't have a T0 to T1 graph to compare it with, but it'd be probably way out here for a month or so.
So is this just for like one certain species, or is this for...
Well, I think Chris is asking, would this be for one species? The species that this experiment was done with was goldfish.
Okay.
But I think the hope is, and I'm not sure it's any more than a hope, is that it applies broadly to fishes. I used the same technique when I was working on my dissertation at Wisconsin to estimate acclimation state in bluegill. And I didn't really do as complete a set of experiments as Brett did, but I didn't see any reason to think that it didn't apply to bluegill. And in fact, what I've done, get out of this and stop drawing. Well, I didn't want to go back all the way to the beginning, but it apparently misinterpreted that. There it is. Okay, so what I did was to actually come up with in order to make a model that simulates a process, you got to quantify the relationships in that. And I guess if I had to defend myself, to all of you folks, both here in the room as well as people who are distant, about why it is that there's so much math in this course, the answer I would give you is that in order to take ideas and make them do anything in terms of predictions that are quantitative, you're going to have to do some math. And that's true in this case too. You got to have a mathematical expression that would account for the behavior that you see in the data. And the relationship that I came up with that I thought explained the data pretty well was one that says that the acclimation state, as measured in this sort of indirect way that Brett measured it by measuring the equivalent state causing the same response, that rate of change in acclimation state is an exponential decay, all right, with coefficient K between the current temperature to which the animal is exposed and the existing acclimation state that it has at that moment. You all by now, I know, would recognize that as an exponential decay process where TACCL is going to approach asymptotically to TA as time goes along. The thing I didn't say is that K here, I said it was a coefficient, I didn't say it was a constant. Because now, unlike the other cases that you've studied of this sort of response, I would argue that K itself is not a constant, but in fact it is itself a function, an exponential function of ambient temperature, where C1 and C2 are such that the value of K changes from being about 2/10 inverse days at the low extreme temperature that the animal can survive, the ultimate lower lethal, to maybe 2 degrees per day per degree at the upper. So what we're saying is the animals acclimate at different rates depending on what ambient temperature they're doing business at, and that's consistent with what Brett's data showed. And I think it's about an order of magnitude difference in the rates, being 2/10 of a degree per day per degree. That's the way we would say an exponential rate constant, right? Degrees per day per degree, or inverse days. to 2 degrees per day at the upper end. People that deal with fish and fish transfers, like my colleagues with Texas Parks and Wildlife and other agencies that are moving fish around in nature and putting them in different habitats, have a rule of thumb that they apply. And that rule of thumb is when you ask them how fast a fish can acclimate to a change, the answer usually comes back, well, about 1 degree per day. In other words, if you move them, it'll take them about 1 day for every degree difference to really get fully adapted to that new world they're in. And I think maybe this is the reason for that, because notice that 1 is right in here somewhere between the 2 extremes. But what you don't get from that answer is that it's kind of like growth rate. Animals don't grow in grams per day, they grow in grams per gram per day. And I don't think fish or any other animal acclimates in degrees per day. They acclimate in degrees per degree per day, meaning that the rate depends on the magnitude of the driving force. How many degrees is different between where you are and where you're going. If it's 1 degree, then it works out to be the same. Degrees per degree per day is degrees per day if the degrees difference is 1 degree. Another way of looking at it is percent per day or proportion per day. So what we'd say here is that animals are acclimating instantaneously at 20% per day when they're adapted to the lowest temperature they can be adapted to. And when they're adapted to the highest temperature, they're actually acclimating at 200% per day, meaning they can more than do it in 1 day. By now, you should be reasonably facile with this idea of an instantaneous rate and converting it to a proportion or a percentage. And so 100% per day comes right in here somewhere intermediate. Notice that this is graphed on a log scale, or maybe you don't notice that, because we're arguing that decay coefficient is an exponential function of temperature. So if you took logarithms, you'd have log of k is C1 plus, or log of C1, you'd have k equals, log of k equals C1 plus C2 times TA. So Ecofish does it this way. Ecofish got to do something. And what Ecofish does is do what I think it ought to do and to be consistent with the observed patterns. And so what we have is experimentally defined rate constants C1 and C2 to work for Ecofish's thermal tolerance zone. And I'm not sure what they are. I think we may find out in this last lab when we do the little application thing at the end, which has to do with how fast do you acclimate and change your temperature preference. We do an application on Lab 5, I think, that deals with that. So anyhow, there's my model. And my model, I believe, produces results that are reasonably consistent with about all the data there are, which aren't very many. I still think Brett's study is probably the landmark study in this area. People are afraid of transient state dynamics. I mean, that's what it comes down to. I think the attitude is, hey, the steady state is so complicated, how can I possibly deal with the transient state? So I'm just going to avoid that and I'm going to have to do everything steady state. Well, that's great, except for the fact that we live in a transient state world. So here's the view that I have of capacity acclimation, and this has probably been more carefully studied by people like C. Lad Prosser and some of the old-timers that did a lot of physiology. And the idea here is that oxygen uptake rate, for example, as a measure of performance, varies with time subsequent to a step change in something like temperature according to a whole series of possible patterns. And you've already encountered these patterns in the steady state. And I said at the time it would be easier to understand these patterns and how they're named and why they're named the way they are if you saw the transient state. And now I'm going to show you what I meant. So let's suppose we're going along here at this temperature to this point in time. The dashed line shows your temperature. And then there's a step change in temperature to a higher value that is maintained for indefinitely long after that step. And then we measure oxygen uptake rate, routine metabolism, let's say. And so it's some level, whatever it is. This is arbitrary. I mean, the scale obviously for VO2 is not necessarily connected at all with the scale for temperature. I'm just trying to show both on the same graph. At this point in time, there's a step change in temperature, and typically animals pick up on that really quick. And the metabolic rate, it's an alarm response, I think. I mean, it just spikes. And it doesn't matter typically whether the step is down or up. It's an overshoot response. It's a spike in metabolic performance or activity. So you get this little overshoot here, or big overshoot, and it persists for a very short time, typically only a few seconds or maybe a minute or two. And then the rate falls back to some lesser value that's intermediate between where it started and the value at the maximum overshoot point. Falls back to this intermediate level. And then it gradually just sort of changes back to a new steady state. Now, the new steady state that it comes to is consistent with the type of acclamation, the type of compensatory change that's demonstrated by that particular species and that particular performance response. If the new acclimated state, the one that you measured days or weeks later after this change, was not very different from the rate that we say is the stabilized rate that we would measure in a few minutes or an hour or two after the change, then we would claim that this system demonstrates no acclimation, because the acclimated state is the same as the stabilized state. On the other hand, let's suppose that after days or weeks, what's happening here is a change in the scale. That's the reason this curve has a different rate of fall from this curve. So imagine the scale from here to this point, from the left to there, is, in minutes or hours. And then as you go from here on out, the scale changes from minutes to weeks or minutes to days or something, hours to days. It changes to a longer interval. That's the reason for the change here. Is that making sense? That's all right. So, but look what happened. In the case of perfect compensation, the new acclimated state finally comes back to the same level that it had before the change. In other words, the intent here is that this level before the temperature change is the same as this level after the change and after days have gone by. So the animal was able to fully compensate for that change in temperature and adjust all of its physiology back, so it was performing after this, at the end of this time, at the same level it was performing before the change. So that's the reason we say it was perfect. But suppose that as we go from the stabilized state to the acclimated state, the response doesn't get all the way to perfect. It's intermediate between no and perfect. We call that a partial acclimation, partial compensatory change, partial acclimation. Now, what happens if the change not only comes to perfect but keeps on going, and you end up with a new acclimated state that goes through perfect and all the way to this point? Well, that's what we call over. And that's why we call it over. It went through the perfect response and all the way on the other side and ended up down here somewhere. Anywhere between perfect and as low as you want to go, we would consider overcompensation. And I didn't show the final member of that set, but you know where it is. You ought to be able to draw it. The final member of that set would start here and go up and end up here someplace. In other words, a curve that's going like Alt Z. In other words, I didn't draw this one, but that would indicate what kind of compensatory response. Reverse. Absolutely. And you can see why it's reverse. It actually went the opposite direction from what you would predict would be the right direction for perfect compensation. Okay, so that's the story of the transient state system. I don't know any. Well, what do I know about this? Do I have it? Yeah, well, it's in EcoFish, and I can't remember exactly how I programmed it. I think what I did was just use something like the resistance acclamation program for an exponential decay process. We maybe could dig inside and figure it out. I probably did that in the wee hours some morning, and was lucky to get it to work at all. I don't remember how I did it. Been a while. So, temperature isn't the only factor that fish acclimate to, and I've made that point already, I think. One of the other factors they acclimate to is photoperiod. And when I say they acclimate to photoperiod, I mean that they make physiological changes in response to the changing day length that are independent of other factors as far as we can control the other factors, like temperature. In other words, you put the fish at constant temperature and expose it to change in day length in the normally, or even an accelerated pace, as we do in maturation work with fed fish, like red drum. They change their physiological states in a way that's consistent with what you'd expect in the changing seasons. They become more heat tolerant when they're exposed to an increasing day length, for example, and less cold tolerant. And there's some funny things going on there that I don't think anybody understands. I mean, there are some changes in day length that result in jumps in the resistance and capacity. It's not a gradual thing always. What they're doing is, as the day length is increasing in early February, jumps all of a sudden, so that by the time you get into March, it's way up compared to what it was at the end of February. And I'm not sure what's going on there. Undoubtedly, the endocrine system, mediated by signals in the form of light, one presumes, through the eye, maybe through the pineal body in the brain, modulate that response. Dr. McKenzie needs to tell us how that works, how photoperiod causes pituitary and thyroid responses. Pardon? I'm sure it does. It must, Something's changing. And EcoFish doesn't have a pituitary or a thyroid, so EcoFish is not sensitive to photoperiod. But we could probably write something, that would make it act as if it knew that it had a pituitary. Low DO is another factor that EcoFish does know how to deal with, a factor to which fish acclimate. And the pattern of response is indicated by the graph there, where if you've got two different fish, or the same fish acclimated in two different ways, let's say, one that's in a tank and been exposed for a long period of time to air-saturated dissolved oxygen concentrations, another that's been in the same kind of situation but exposed to only 50%. The pattern that I would expect is that oxygen uptake rate, and here I'm talking, I guess I'm talking standard rate. I don't label it, but I think I'm talking standard rate. The rate is reduced through the zone of independence, that is, where DO is out here on the right and the line is flat. The rate tends to be lower if you're acclimated lower. You don't have as much demand for oxygen if you aren't accustomed to as high an oxygen level. And then you hit the wall at a higher value if you're acclimated to air-saturated water versus oxygen-deficient water. And then finally, the lethal level maybe I'm thinking about routine rates here, because as I told you guys over and over, I've tried to convince you that lethal is the same as the critical, basically, if you go time independent. So maybe I'm thinking about routine levels. So you can see this line here. They're on the same line, and it's sort of going to the point 0, 0. And later on, maybe already, I have labeled the slope of that line, the marginal metabolic scope. That's the ratio of rate to the limiting value for that rate. And what we're saying is the limiting value for air-saturated acclimation is higher than for the reduced acclimation value. But they're on the same line. There's, in both cases, this little hump, and we talked about the little hump and why it would be there, and maybe it's got to do with an alarm response, and maybe it's an artifact of doing respirometry in little containers where there's a probable feedback that's real quick between the oxygen uptake rate and the amount of oxygen in the water. And the faster your rate, the more oxygen you use. So the faster the rate, the faster the value of DO drops in the container. Well, that wouldn't be true in a real open system. The rate of the animal's metabolism would have almost nothing to do with the DO in the water, because that one animal is not going to be able to have much of an impact. But in a little chamber, in other words, I think that hump is an artifact of respirometry, the way humans do it, not the way fish do it. So 3 things happen, none of them particularly good. If you're acclimated to low DO, you lower your metabolic rate in the zone of respiratory independence. The limiting value is lowered to a lower DO, and the lethal value is lowered maybe. So the end of the line comes at a lower DO if you're acclimated to low DO. In other words, you're better able to withstand low DO if you're acclimated to low DO. Some new information from one of my recent PhD students, which is getting ready to be published, I hope. He's already published a couple of papers, but he hasn't published this one. And it says that shrimp, at least Penaeus vanami, or Litopenaeus, I guess it is now, may not demonstrate any acclimatory response to DO. That finfish regularly do, but shrimp may not. And this suggestion was from Scott's other co-chair, Dr. Lawrence, Addison Lawrence in Port Aransas, who said that he had noticed that if you take a shrimp from low DO water, and it's been there a long time, you take another shrimp from high DO water, it's been there for a long time, put them in the same water, they perform the same way. In other words, looks like no compensation. It's not a benefit to be acclimated or to be held at low DO if you're a shrimp, because when you're put in higher DO water, you perform better, but then so does the shrimp that was not acclimated. Both perform the same way. In other words, these 2 lines for shrimp look like they're in the same place. They're on top of each other.
What was low DO for them?
I don't know. Alejandro says, what was the low DO? I think Scott's work may have dealt with 50% air saturation. I can't recall exactly. I got his dissertation somewhere on my computer. We could look, maybe. Hopefully we'll be able to see it in print in the public forum, a journal at some point in this near future. But finfish, I think, do acclimate. You take a fish that's sort of lived the life of a couch potato and lived the life of Riley, luxurious, plenty of oxygen. You put it in a bad situation, it just crashes. You take the same fish, and it's been adapted, it's been, toughened up by its exposure to tough situations, and it's more tolerant of hard situations in terms of low DO. That's what we mean by acclimation. Compensatory changes. Fish do make compensatory adjustments to salinity. And it's pretty obvious, but you know what? Ecofish doesn't. I just haven't built it in. It's just one of those things I never got to. This is obviously important, especially with estuarine fish that see a variety of salinities, maybe over in a short framework in terms of time, maybe a few hours. You know, mullet moved back and forth in the estuary from nearly fresh to full strength or even hypersaline water over a period of a few hours. So undoubtedly they're doing some interesting things. Personal story, you know, it always helps to have a little story. When I was with the Fisheries Service in Honolulu, we got the bright idea that tilapia might make good chum for skipjack tuna because sometimes it was really hard to come by these little native anchovies, the nehu, that the fishery caught in Pearl Harbor. That was the best place for getting them. Sometimes you didn't have many, you know, population would be low. So we thought we would try tilapia. And there was a population of, I guess it was tilapia, I'm going to say melodica, but maybe zelai, one species or the other, not aurea, in this little freshwater reservoir. So we went up there and caught a bunch of these things and took them down to Koala Basin and put them into one of our holding tanks, which was supplied with fresh seawater straight out of the ship channel. And boy, they all just died in about 30 minutes or a few hours. So then we thought better of it and we tried again. And that time we put them into a tank that had, I think, a third strength seawater, diluted by a factor of 2 to 1. And they lived fine. And then a day after we put them in there, we raised the salinity by changing the valve settings so it went up to 2/3. So first maybe it was 15 or 12 parts per thousand. Then it went to 20 or 24. And then it went to 35. So we did that in a 3-day, and the fish were fine. So they could stand the challenge if it was a gradual thing. You know, they were presumably given time to do some compensatory adjustments to the changing salinity regime. So what's the punchline? Did it work? No, it didn't work. The fish did fine. I mean, they survived once we gradually changed salinity. We took them out into the ocean off Hawaii in a holding tank supplied with running seawater, same way we would do nehu, bait tank. And then we got into a bunch of feeding skipjack. We tried to chum by throwing these tilapia overboard the way we would do nehu, and it didn't work. Why didn't it work? Well, we got a diver over the side, and he said what happened was these tilapia were smart enough that they just formed like a second skin right around the hull of the fishing boat. They just plastered themselves right up against the hull, and they were so thick that they looked like a second skin, and the tuna didn't see them as food. Whereas the nehu would go out and do the typical panic thing, and boy, that would just bring on the feeding frenzy. So it was an interesting experiment, even though it didn't say much about, that's aside from salinity, I guess. We tried hauling a different anchovy from Hawaii or from California, and that didn't work so well either. That turned out to be a pretty expensive deal. Toxic substances also generate inflammatory responses in fish, and you'd ask yourself, well, why in the world would synthetic detergents or phenols that fish have never seen until humans came along, why would they why would there be any adaptive response to something that exotic? And the answer seems to be in every case, nearly. Well, what these things are really doing is interfering with respiration, either respiratory exchange at the gill or transport of oxygen in the hemoglobin, or it's basically operating like oxygen insufficiency. So ecofish acclimates to ammonia, and we did that intentionally so that if people wanted to explore what happened if you preconditioned them to ammonia, you could do the same experiments we did in the lab. What was it, lab 2 or 3? 3, I guess. And ask the question, how would ecofish respond to the same levels of ammonia given that it had prior exposure to sublethal levels? Or copper. There's also an inflammatory response to copper. So some of you who are interested in toxicity bioassay might be interested in a project that would focus on acclamation. One thing one might do is generate a temperature tolerance polygon for ecofish by doing step change experiments and looking to see what steps result in incomplete mortality after 10,000 minutes or 96 hours or whatever, and which ones don't. What defines the set of all possible combinations of acclimation and test temperature that lie within the zone of tolerance versus the set that lies outside? So how can fishes have evolved mechanisms? I just tried to answer that by talking about the fact that these toxic effects get translated into a mechanistic effect that masquerades or looks like something else, like suffocation. I think that's how. What are the mechanisms? And boy, that's a can of worms. What mechanisms are involved in these compensatory changes? Some are pretty obvious, like low DO, it looks like, and anything that triggers oxygen stress looks like it's typically triggers increased hematocrit. So there's just more hemoglobin, more red blood cells circulating and more hemoglobin. The character of the hemoglobin molecule itself, the type of hemoglobin, may also shift in response to acclimation. Low temperature is regularly, adaptation to low temperature, as I told you way back there when we were talking about environment, is regularly characterized by an increase in the degree of unsaturation in the fatty acids, particularly the ones that are particularly important are the ones in the phospholipid membrane, cell membrane, not so much depot fat. I mean, it wouldn't matter there, but in the membranes. Increased unsaturation of the fatty acids in the fats that are there correspond with increased fluidity at low temperature. So that's obviously a good mechanistic connection. I mean, you want your membranes to continue to operate at low temperature with the transfer of stuff. And so it would help if you lowered the membrane fat melting point. And that's what having a higher degree of unsaturation tends to do. There may also be some reduction in fatty acid chain length, which also tends to increase fluidity. High temperature adaptation is regularly associated with decreased water content of the tissues. And again, I guess that makes sense because if there's less water in there, then it's more of a solid and less of a liquid, so maybe the diffusion rates are slowed down, and maybe you don't want to have it diffuse as fast. If you want to compensate for a real high temperature, you don't want to let things run away with you. So people, expose fish to low temperature, high temperature, and then measure everything they can think of. And one of the things they measure is the water content of the tissues, and it typically is less. There's dehydration when you acclimate to high temperature. In general, I guess, there are molecular and cellular changes that promote homeostasis of one kind or another. And there's some, new work that I don't understand very well, so I don't talk about it much, involving things like heat shock proteins, which are little proteins that affect the conformation of other proteins. And, in looking at the literature, and I would like to know if there's anything recent that changes this, but it looks to me like heat shock proteins mainly have been used simply as markers of the fact that an animal has been stressed at some point in its recent past by temperature or by something else, because other stresses can also result in the formation of these so-called heat shock proteins. The P450, the cytochrome oxidase system, that's an enzyme that's really important in cellular metabolism. I guess the the P is probably for pigment, and the 450 is the wavelength of light that it does something at, fluoresces, absorbs. Anyhow, the P450 system characterizes animals that have been exposed to toxic substances like heavy metals. So you can use them as markers, and find out whether or not your fish has had a tough recent life, in the slums, being exposed to heavy metals or something. I've been involved in some research that we did with colleagues in the vet school on that at one point and published. But to my knowledge, there's not any good mechanistic attempt to connect the existence of these heat shock proteins or the P450s with mechanistic change in heat tolerance, for example. It's more an indicator that there's been some damage. The one thing that was kind of impressive, but I think it's losing favor, has lost a lot of favor, was this isozyme theory of Peter Ochachka, who was at the University of British Columbia for many years and a highly regarded physiologist. And what Ochachka showed was that when you adapt fish, and these were all salmonids, I think rainbow trout, to high and low temperature, that the reaction kinetics of important enzymes tends to shift to different patterns. So rainbow trout acclimated to low temperature have a Michaelis-Menten coefficient here, a Km, that reaches its maximum activity, its lowest value. In other words, the substrate has to be the substrate can be at the lowest concentration at that temperature and still get the same reaction rate. In other words, these things are really... maybe I should have just graphed this as activity and just done the inverse of the curves so that the peaks were up. Anyhow, this is the best temperature if you're acclimated to a low temperature, 5 or 6 degrees. That's where Km is minimum for that particular enzyme. And if you're acclimated to high temperature, then you do better up here around 18. So that's clearly adaptive, If you acclimate to high temperature, that shifts the form of key enzymes to their variants that perform better at high temperature. And similarly, if you shift it low, then the low. And what's the problem with this? Well, one big complaint people have made is that rainbow trout are natural tetraploids, and so they might have more of this kind of machinery than other fish. And other fish don't show the existence of isozymes for the same enzymes that rainbow trout do. So there's been, I guess, a lot of questioning about this idea, and I'm not sure where it stands at present. I always thought it sounded neat, but I don't know enough about it to be very critical. So I believe that might be the end of the show for Acclamation. I'm going to pause and change shows. Okay, I took the opportunity while we were paused here to share some information with the local group that I'll now share with everyone, and that is that I might just ordinarily terminate here. We've got about 15 or so minutes of class time left, but I want to push on and cover as much ground as possible because I'm trying to actually get far enough ahead that we could maybe have a break from the lecture session a little later on. Everybody at this point in the semester needs all the breaks they can get probably to catch up on other things. I do too. So anyhow, I want to begin the consideration of perception today, and we'll just see how far we get. The context of this perception, what I'm doing now is I'm getting ready to head into a consideration of behavioral responses that complement physiological responses. And I want to, concern myself a little bit about how it is fish detect the variation in environment, how they perceive environment. So up to now, we've talked about environment and we've talked about what I would call responses in place. So you take a fish and you put it in a jar or a tank and you pass the environment through the tank or the jar, and it has to deal with whatever you give it. It can't choose. So these amount to exchanges that go on between the fish and its outside world. And all the fish can do is regulate its internal environment, try to hold things in there in a more favorable distribution than what goes on outside. We call that physiological regulation of internal environment. Once the capacity or once the regulatory abilities are exceeded, then all you can do is change over the long term and achieve maybe some acclamation. Either capacity acclamation that improves your ability to perform whatever it is you need to do, or at least extends the time that you can live given that particular disturbance, in hopes, I guess, quote, that the disturbance will come to an end or the stress will come to an end and you'll be restored to better times and places and things and you won't have to deal with it forever. So that's the reason that resistance acclamation is so important, is that it does extend the animal's life possibly beyond the time when the stress is going to be imposed. If you had to live forever at that new level that's going to be lethal, why would it matter if you could live a day or a week? Maybe you'd like to live a week if you could, but you're going to die eventually. So the idea is here is that lethal environmental situations hopefully don't persist forever. They come and go. So it's important to be able to get through those and come out on the other side, and then have a better world to live in. There's a whole other set of responses that I will call, as opposed to responses in place, these are responses that amount to moving around to a different place, distributional responses. And we're going to deal with those today in Lab 5. That leads us to what I call behavioral enviroregulation. Behavior that somehow serves to regulate the total complex of surrounding environment so that you don't have to deal with all the stresses and all the insult of really an abnormal environment if there's some better place to be in the system. People who work on terrestrial animals tend to think of this more as habitat selection, because they're used to thinking of habitat as being distributed in space and animals moving from one part of the space to a different part, so they're in a different habitat. So you're selecting habitats. In the case of fish, where a lot of the important properties of the world are things that are in the water and are moving past and they're ephemeral, then I don't think selection of habitat is what's going on. It's actually selection of environment within, wherever you are in the habitat. So that's the reason I call it behavioral enviroregulation rather than habitat selection. And to some extent, but not necessarily completely, but to some extent, that depends on being able to perceive the variation in environment from place to place and time to time. And that's what I want to focus on here. And I want to walk through the factors that we've talked about all along as being really important to fish. The first of these being temperature. We know more about that factor than almost anything else. I think because it's easier to measure, mostly, and because it is so important. And fish are really good at detecting little changes in temperature. Clever experiments have been performed. The first by a guy named Bull, H.O. Bull, B-U-L-L. And what he showed was that fish can detect changes as small as a few hundredths of a degree when the change is abrupt or rapid. Say when the change is at least a degree per minute. How did Bull find that out? Well, he did some clever experiments, as I said, that we call conditioning experiments, learning experiments, threshold discrimination experiments. And it involved training fish to respond to small changes in temperature that they could detect. Normally, fish don't respond to these little bitty changes because they're beneath their notice, so to speak. Beneath their notice. You know, you and I are the same way. We walk into a room, the temperature is 1 degree different. We don't pay any attention. We don't even notice it. We walk into a room, the temperature is 20 degrees different. You probably notice it. You probably go, whoa, and respond in an unconditioned way. But if somebody told you, hey, $100 riding on whether or not you can detect the change in temperature when you go from the hall into this room, you might be a little more sensitized. That's the whole idea of conditioned responses. It is working to make it worth the animal's while to communicate to you, the researcher, that the animal has noticed something. And what H.O. Bull did was work to make it worth the while of fish in a tank to communicate the fact that they detected small changes that Bull imposed on the temperature. He did conditioning experiments. And what he actually did was he had these fish in a raceway in I think like a Quonset hut type building. Water flowing through it. Variety of fish. And there was a pipe bringing water to the raceway from outside the building. And every once in a while he'd have his technician sneak out there with a blowtorch and apply it to that pipe. Metal pipe. And heat the water up. Coming through the pipe a little bit, a pulse, and then let that flow down into the raceway. And once he was confident that that water had hit the raceway and the fish had been exposed to that increase in temperature, he had the technician send some feed down another tube, a sort of a feeding tube, and feed the fish. So the conditioning stimulus was the temperature change. The reinforcement was the feeding. He used positive reinforcement. Other people have tried to do conditioning experiments with negative reinforcement. You don't detect it, you get punished. And you know what? Fish are, I think like people, they'd a lot rather have positive reinforcement. And usually the negative reinforcement that I've seen applied usually just serves to frustrate everybody, the animal and the experimenter. But, after a while, what happened was Bull would send that pulse of heat down the tube, and the fish would come up to the upper end of the raceway and start going into this food search behavior. And you could peek through the hole in the peephole and see that. Even though there was no feed, so they were anticipating the reward they were going to get because they had gotten a signal. The signal that the reward is coming. So he probably frustrated them right there that one time. But then they had fed him after they showed him the reinforcement, so he didn't allow the behavior to be extinguished. After a while, animals will extinguish behaviors that aren't reinforced. So the fish communicates to the observer its detection of a stimulus by some change in its behavior, moving to the feeding place or starting to salivate if you're Pavlov's dog. Same idea. But you know what? Pavlov was just working with learning. He wasn't working with discrimination as far as I know. He didn't try tinkling that bell lighter and lighter and lighter. He just rang the bell, fed the dog. Rang the bell, fed the dog. And after a while, he rang the bell, the dog starts slobbering. It's expecting to get fed. But he didn't ask, can you hear the bell? He might have, I don't know. But Bull and his technician did ask the question, can you detect this temperature change? So he would tell the technician, lay that heat on heavy, get it going, get the fish conditioned. Now back off and just give a little bit of heat. And, his thermometry wasn't real good, so he wasn't sure exactly how much temperature the fish were getting, but he was calculating that they were getting a few hundredths of a degree. And as long as they got just a few hundredths, they reacted. They could detect. They demonstrated, they communicated by their behavior that they could detect the stimulus when it was as small as just a few hundredths of a degree. Well, long after Bull came John Bardock at the University of Michigan and a guy named Bjorklund, and they did a much more sophisticated confirmation of Bull's experiments. They had fish in aquaria, and they had very sensitive thermometers, and they did the same kind of conditioning work. And they had a neat control in that they not only, there was always complaints about Bull's work. Okay, well, they didn't really detect the temperature change. What they detected was the lead that got sloughed off the pipe because you heated it up with a blowtorch, or they detected the clink when the guy accidentally tapped the pipe with the blowtorch, or, there were all these confounding things that might have gone on. So what Bjorklund and Bull did that was so clever is that they used both temperature increases and decreases. So, hey, that takes out the Arrhenius effect. Because, maybe you could detect the higher temperature and you'd respond more because the temperature's higher. Well, what if it's lower? So what they showed was, first of all, that the same range of threshold values that Bull had reported was relevant. Not only that, they showed that fish could be trained to respond to increases in temperature and ignore decreases, and vice versa. So that was really pretty much conclusive experiments. It eliminated a lot of the concerns about Bull's earlier work. So those are conditioned responses. The responses, thresholds for response where you're making it worth the animal's while to detect anything it can detect. What about unconditioned responses? How much does the change in temperature have to be when you walk in the room before you show some response without being conditioned, without being given any kind of reward other than from a regulatory, response itself? And the answer there for fish seems to be about one order of magnitude greater than for the conditioned response. In other words, if the conditioned response is 0.03 of a degree, then it may need to be 0.3 of a degree. Or if it's 1/10 degree, then maybe it's 1 degree. So I'm saying that thresholds for unconditioned response to abrupt temperature change in fish is around 0.5 to 1 degree. And these responses typically involve preference or escape behavior. In other words, temperature has become important for its own sake, as opposed to being, a signal that something else that is important is going to happen. Do you understand the distinction there? In the case of conditioned responses, temperature is a conditioning stimulus. But in the case of unconditioned responses, temperature is its own reward. It's the temperature itself that's important, the change. I already told you temperature increases are discriminated from decreases. What I haven't told you is that other work that came along about the time of Bardach's work and Bjorklund's work showed that there was a lot of debate about, where are the receptors? Where are the anatomical devices? And the answer seems to be that there aren't any. It's just generalized dendritic free nerve endings in the skin that are the temperature receptors. A lady named Charlotte Sullivan at the University of Toronto did some clever experiments. She dipped fish in procaine. She deadened their skins, and they couldn't figure out where they didn't care. No, they couldn't respond in a temperature gradient. Whereas control fish that were spinalectomized, having their spines severed just behind their cranium, would wobble their way and manage somehow to get themselves into the preferred temperature environment. So spinalectomy, that was kind of a good control, I thought, because if you can do it without your whole posterior parts working, but you can't do it if your skin has been made unresponsive. Well, okay, so there's another set of very clever experiments done by whom? The name will come to me in a minute. I don't have it written down. Maybe I got it somewhere in my notes here. Larry Crawshaw. It came to me already. Out at Stanford. I guess he was at one of the California schools anyhow. Actually, Scripps, I think. Anyhow, what Crawshaw did was he was interested in whether or not all the thermal signaling is coming from these skin receptors or is some of it coming from inside the fish. Deep receptors, he called them. And what he finally concluded is that peripheral inputs from these free nerve endings in the skin are compared neurologically with inputs from deep receptors in the brain, maybe in the midbrain of the fish. And that provides the fish information about the direction of environmental temperature change through time. And I'm going to explain how that works. Ecofish does this, by the way. So the deep receptors are greater than the skin receptor temperatures if the fish is cooling off, because those deep receptors are remembering, so to speak, a past environment more than the skin receptors. The skin receptors have very short term. They're exposed right now, this moment, to temperature, whereas the deep receptors are giving you values that were reflecting the past exposure. This leads to what we call the thermal lag hypothesis of gradient perception. We've sort of developed this to its greatest degree for tunas. And the argument goes like this: if you're going along and the temperature is flat, then the body temperature and the ambient temperature of the animal, the body temperature of the animal is about the same as its ambient, maybe just a little offset by this equilibrium excess that you know about now. But now if it enters a temperature gradient where ambient's declining, and the fish has any thermal inertia at all, which all fish do, the bigger they are, the more tuna-like they are, the more the thermal inertia, as you learned if you did the little added value thing in the last lab, then the differential here between the body temperature that exists at every moment and the ambient temperature at that moment gets greater. And not only that, it tells you the direction of change, because if body temperature is higher than ambient, that means that you're coming from a warmer place to a cooler place. And I didn't draw the other graph, but you can draw it for yourself. Suppose the ambient had gone up. Body temperature lags behind. Now you're coming from a cooler place, and body temperature is behind, lower than ambient at the moment. So the maximum value of the absolute difference between body and ambient is going to be, it turns out, the rate of temperature change with respect to time divided by the rate coefficient for heat transfer, K. We're going to quit there. We're at 12:25. Kind of relevant because we're going to deal with some of this in the lab today, some of the consequences of this thermoregulatory behavior. Anybody have anything to say before I punch the pause button? Okay, we'll get back together then, maybe be joined by some additional folks at 2.